Now let's discuss synthesis of glutamate cotransmitters glycine and D-Serine. Glutamate systems are curious in that one of the key receptors for glutamate requires cotransmitters in addition to glutamate in order to function. That receptor is the NMDA receptor and the cotransmitters is either the amino acid glycine or D-Serine. The glutamate neurons get the glycine they need for the NMDA receptors either from the glycine neurons or from the glia. Now the glycine is unable to diffuse very far from the neighboring glycine neurons as they are taken back into those neurons by a type of glycine reuptake pump known as the type 2 glycine transporter or GLIT2. Thus, the neighboring glia is thought to be the source of most of the glycine available for glutamate synapses. Now this glycine can be taken up into the glia as well as the glutamate neurons from the synapse by type 1 glycine transporter or GLIT1. It can also be taken up into the glia by glial SNAT. Interestingly, it is not known to be stored within synaptic vesicles of glia. But this glycine in the cytoplasm of glia is nevertheless somehow available for release into the synapse, and it escapes these glial cells by riding the aforementioned GLIT1 transporter. Thus, the GLIT1 transporter transport glycine both in and out of glial cells. Once outside, the glycine can get right back into the glia by an inwardly directed GLIT1 and is thought to be the main mechanism responsible for terminating the action of synaptic glycine. But in contrast, D-Serine is thought to be stored within some type of synaptic vesicle within the glia. There are novel treatments for schizophrenia that boost glycine action and thus glutamate action at NMDA receptors. And these include inhibitors of key glycine transporter GLIT1 and selective glycine reuptake inhibitors or SGRIs. Glycine can also be synthesized from the amino acid l-serine, transported into the glial cells by an l-serine transporter known as l-sert, and converted from l-serine into glycine by a glial enzyme known as serine hydroxymethyl-transferase or SHMT. As can be seen by the equation, this enzyme also works in both directions, either converting l-serine into glycine or glycine into l-serine. Now D-Serine is very unusual as the 20 known essential amino acids are all L-amino acids. It just so happens that D-Serine has high affinity for the glycine site on NMDA receptors and that glia are equipped with an enzyme called D-Serine racemase that converts the regular L-serine into the neurotransmitter amino acid D-Serine and vice versa. Thus, D-Serine can be derived either from glycine or from L-serine. Interestingly, the D-Serines so produced may be stored in some sort of vesicle in the glia for subsequent release on a reversed glial D-Serine transporter or D-Sert. Now D-Serine's actions are not only terminated by the synaptic reuptake where the inwardly acting glial D-Sert but also by the enzyme D-amino acid oxidase or DOA that converts D-Serine into the inactivated hydroxypyruvate. Now brain makes an activator for DAO known not surprisingly as D-amino acid oxidase activator or DAOA. The gene that makes DAOA may be one of the most important regulatory genes that contribute to the genetic basis of schizophrenia and thus is a part of the neurodevelopmental hypothesis of schizophrenia.